Hello and welcome to Nikolai's genetics lessons, and today's problem is tall plant height, capital T, and the P is dominant over the short plant height, which is small t. Question A: if true breeding tall plants are crossed with true breeding short plants, what genotypes and phenotypes would you expect to see in the F1 generation? So, in order to answer this question, you have to know what is true breeding means. So basically we have here two alleles, dominant allele T and recessive allele T. And in diploid organism, these two alleles can produce three different genotypes. One is homozygous dominant, another heterozygous, and the last one would be homozygous recessive. Out of these three, this one and this one we call true breeding, because if we cross plants with itself, so this genotype with the same genotype, all the progeny would be of the same genotype and phenotype. And the same is true for this genotype. If we, for example, if this is going to be a plant and if we pollinate it, self-pollinate, then 100% of the progeny also going to have the same genotype and phenotype. But if we cross this plant with itself, we would see segregation of trait. 3/4 would be phenotypically tall and 1/4 would be phenotypically small. So only these two genotypes we call true breeding. So according to our question A, true breeding tall plants, and we know the genotype of the true breeding plant is capital T, capital T. We have to cross with true breeding small plant, whose genotype also is known, and which is small t, small t. And now if we build a Punnett square, we can see result of such a cross. As you see, 100% of the progeny going to be heterozygous and phenotypically going to be tall. So once again, these two genotypes would produce one phenotype, which tall plants, and this genotype would produce one phenotype, which is small plants. And as you see, according to our Punnett square, 100% as a result of such a cross of these two parents would be plants which is going to be phenotypically tall. So when we cross one true breeding tall plant with another true breeding phenotypically small plant. Now question B: if F1 plants are selfed and 1000 F2 plants are obtained, how many of these F2 would you expect to be true breeding when selfed? And once again, this is going to be our F1 generation, and if we cross two plants from F1 generation, so here is the genotypes, what we are going to expect in the following generation, which is going to be F2 generation. Capital T here, capital T, small t here, capital T, small t here, and small t here. And here we have one genotype that is true breeding, and here's another genotype that is also true breeding. So we expect that 50% as a result of a cross two parents from F1 generation would produce genotypes that we call true breeding, or 50% of the F2 progeny would be true breeding. And question C: a tall plant of unknown history is test crossed and the resulting progeny are all tall. Draw the cross, including the most probable genotypes of the parents and offspring. So what do we call test? We test with a plant of known genotype and known phenotype. For example, if we would take a tall plant, can we tell for sure the genotype of tall plant? It can be homozygous dominant or it can be heterozygous. So phenotype is one, but genotypes can be different. So we're never sure when we take a tall plant what genotype it is. But this genotype produce one phenotype. So if we take a small plant, we know for sure that genotype is small t, small t. So we use this plant for testing. So we have to cross plant that is homozygous recessive with unknown plant, and result would be all plants that is also large or tall plants. So genotype of unknown have to be homozygous dominant. In this case, 100% result of such a cross would be plants that is going to be heterozygous and going to be all of them tall, phenotypically tall. And once again, this is phenotypically small plant, and this is our unknown tall plant. And as a result, 100% of the progeny in such a cross would be tall plants. But what if our unknown parent is heterozygous? And this is exactly next question. If the tall plants is known to be heterozygous, what phenotype ratio do you expect in the progeny? So, once again, let's do another test cross. So we take one plant with known genotype, that is a small plant, homozygous recessive, and now let's assume that another plant is heterozygous. So how this is going to affect phenotypes and genotypes in the next generation. And we are going to get here capital T, small t, capital T, small t, small t here, and small t here. And as you see, we took tall plant, we cross it with a small plant, and in such a cross we expect that 50% going to be phenotypically tall and 50% are going to be phenotypically small plants. So when we do a test cross of known genotype with unknown genotype but known phenotype, two outcomes can be whether all plants would be tall, and we know instantly that genotype of unknown plant is homozygous dominant. But if a result would be 50% of the progeny tall and 50% of the progeny small, then we know that unknown plant, known phenotype but unknown genotype, is heterozygous. And this is what we call test cross. This to Punnett Square is example of the test cross. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any. Share this video with your classmates, and see you in the next video. Goodbye.